Okay, so let's finish the rest of our notes. So the ribosome is the molecular machine that builds proteins using the mRNA instructions. The ribosome is made up of ribosomal RNA, okay, and proteins. It has two subunits, one large and one small. It has three sites, A, which stands for amino acid, P, which stands for peptidyl, and the E, which stands for exit sites. So what exactly happens? In the initiation step, or the first step, the ribosome subunits come together at the 5' of the mRNA. It moves along the mRNA until the A site reaches a start codon. What's our start codon? We know that's AUG. A tRNA with a complementary anticodon enters the A site. So you can see here that the mRNA is going through the protein. Think of it like a machine that's reading. It starts at the G cap. It finds the AUG. AUG is the start codon. When the tRNA finds the anticodon here, the anticodon has is on the tRNA, and that tells it which amino acid to then put together. Okay, so then let's look at the next step. So that's just reading the start codon, the first one. Now what? So that was here. Start codon. This is the anticodon. This is the tRNA. This is now the amino acid that is put together because of the start codon. And this was the G, the 5' that starts. It's like it's feeding through a machine. Now, elongation, which is the second step of the process. The first tRNA moves to the P site. The tRNA with a complementary anticodon to the next codon enters the A site. A peptide bond, those are bonds between amino acids, forms between the amino acids at the P site and the amino acid at the A site. So look here. We have, remember, our 5' starts. It first came through. It read the first start codon. It read the start codon. It hooked up with its anticodon, and the amino acid came from that. Then it reads the next codon, CCU. CCU's anticodon is GGA. How do you know that? Because of the complementary base pairs, G and C, A and U in RNA. And then, because GGA codes for proline, that's our next amino acid. Now, this amino acid and this amino acid are hooked together by peptide bonds. Okay? And it keeps going. So then this would go through and it would read the next one and the next one and the next one until it got to a stop codon. I see the stop codon possibly here. Where does translation take place? So translation itself takes place in eukaryotes. Translation takes place in the cytoplasm where ribosomes are. Remember, it can't do this in the nucleus because there's no ribosomes there. So it makes the copy in the nucleus and edits the copy, but then if it wants to translate the copy, it has to send it outside the nucleus to the ribosome to be read. The ribosome is like a translator. In prokaryotes, it's a little bit different. Translation can take place at the same time and place as transcription. Let's move that up so you can see. Near the DNA because there is no nucleus. Okay, next page. So the elongation continued. Remember, we already did the first 2 amino acids. We're going to keep going. So the tRNA with the 2 amino acids attached moves from the A site now to the P site. The first tRNA that was in the P site moves to the E site and exits. See here, it breaks off. This tRNA anticodon piece breaks off, and the methionine is still attached to the proline amino acid. A tRNA that is complementary to the third codon enters the A site. Remember, so this is the A site here, this is the P site here, and this is the E site here. Okay. A peptide bond forms between the amino acids attached to the tRNAs in the A and P sites. This process continues until the stop codon enters the A site. And remember, we have 3 options for stop codon. We have UAG, UGA, and UAA. Those are your stop codons. And finally, the termination step. So also up here you can see this next codon codes for arginine. Arginine is then attached by peptide bond to proline, and now we have a 3-chained amino acid for now. It keeps going until it reaches that stop codon. Okay, so the termination step, or the last step. So when a stop codon reaches the A site, the ribosome stops translating. Remember, it's the punctuation. It tells it that it's over. It's done. It's the period, exclamation point, question mark, whatever. It's the punctuation. It ends the sentence. The ribosome's subunits separate, and the tRNA separates from the peptide that has formed. Remember, a peptide is named for a protein. So now we have these amino acids all hooked together. We get to, notice here, here's the UAA. This is our stop codon. It doesn't code for an amino acid. It just tells the ribosome to stop. And at that point, this tRNA breaks off, and now we have this peptide here. Okay? So what happens next? The mRNA can be reused by the ribosome again. The ribosome can also be reused and start translating another mRNA. The tRNA can pick up more matching amino acids. The polypeptide, which is our protein, poly means many, peptide is protein, that's the long string of amino acids, moves into the rough endoplasmic reticulum for editing, processing, and sorting. So now that protein can go on to do the job that it needs to do for the cell. Oh, might help if you could see any of that. That's fantastic. So let's go through it again. The mRNA can be reused by the ribosome again. The ribosome can be reused and start translating another mRNA. The tRNA can pick up more matching amino acids. The polypeptide moves into the rough endoplasmic reticulum for editing, processing, and sorting. All right, so those are the rest of your notes. You now have DNA replication, transcription, and translation notes all completed for this second half of the unit. Last week we did the cell division of mitosis and meiosis. So make sure you're going over all your notes. Your homework is up on Google Classroom for today. There's 3 pages. So hopefully, if you have any questions, you can message me either on Google Classroom or through Remind app, or you can send me an email. Hope you have a great day.